Hello, my name is Brittany Harlow, and I'm a research animal scientist with the USDA-ARS Forage-Animal Production Research Unit in Lexington, Kentucky. Today, I'm excited to be here with you virtually to share an update on our forage livestock research program. The mission of our unit is to improve productivity, profitability, competitiveness, and sustainability of forage animal enterprises within the transition zone of the U.S. We're made up of 5 scientists of varying expertise, and each scientist has a technician. Here's some pictures of our scientists at work. So there are only 5 of us, so we rely heavily on our collaborations to work on our mission. We're co-located on the University of Kentucky campus, so we work a lot with faculty in the College of Agriculture, Food and Environment, but also across campus. We work with Cooperative Extension Service, so through ag agents across the state, and also the producers in generating ideas, but also conducting research on their farms. And then we also have a good relationship with the Kentucky Beef Network, and we do pretty regular research on their demonstration and learning farm in Owenton, Kentucky, Eden Shale Farm. So probably the primary condition that we're tasked to work on is fescue toxicosis. There are 86 million acres of tall fescue in the United States, most of which are toxic and can cause fescue toxicosis. Most of the tall fescue is present within this fescue belt. You can see Kentucky's right in the middle. Kentucky itself has about 5.5 million acres of tall fescue. Economically, fescue toxicosis costs the U.S. beef industry about $1 billion in lost production annually. So how does it work? Tall fescue is commonly infected with an endophytic fungus that produces chemicals like ergovaline. These chemicals impart things like insect tolerance and also combat disease in the plant. However, when the animal consumes these chemicals, it can cause vasoconstriction by acting on the blood vessels. So the most common impact of vasoconstriction is reduced heat dissipation, and that can cause an animal to not be as heat tolerant as a normal animal when they're suffering from fescue toxicosis. In this picture on the left, you can see a common appearance of a steer suffering from fescue toxicosis. He's standing in the mud trying to cool off, but also you can see this retained winter hair coat. On the right, you can see more extreme cases in which we'll see things like fescue foot, lost tail switches, and also lost ear tips because of this vasoconstriction. Research has shown that fescue toxicosis can inhibit nutrient absorption, and then there are a lot of reproductive effects associated with fescue toxicosis. So historically, the suggestion was usually to interseed red clovers or other clovers into pastures to combat fescue toxicosis. And the reason for that was that they thought it was diluting out the compounds by giving the animals something else to graze. So why do we use clovers in pastures? Well, they reduce nitrogen inputs required. They can also improve pasture quality by having higher protein and digestibility, increase forage diversity, and altogether that improves animal performance. However, our research group has identified another advantage of red clover and other legumes, and that's that they're rich in isoflavones. Isoflavones are a group of compounds in the flavonoid family. They can act as antioxidants, estrogens, which makes them a little bit concerning for use in breeding stock. But we've also identified new benefits of isoflavones like growth promotion and vasorelaxation, which is relevant to fescue toxicosis, and that's what we're going to be talking about today. So first of all, you can see in this picture 2 images, and these images were taken by a Doppler ultrasound on a goat. The first goat over here was receiving alkaloids with toxic tall fescue seed, and you can see that the goat, this carotid artery is quite constricted compared to the control animal. And the isoflavone metabolites in red clover are vasorelaxants, so they cause the animals that are consuming seed to look more like this control or vasorelaxed. So the first question we wanted to answer is, does it work? So for this experiment, we used Promensal, which is a human postmenopausal supplement, and we dosed that to goats that were receiving a toxic endophyte-infected tall fescue seed challenge. So here we started the fescue alkaloids, and you can see that the goats were all very constricted, which is what we wanted to see in terms of inducing fescue toxicosis. However, when we added in the Promensal or those clover isoflavones, we saw that the animals dilated and were able to get back to this baseline here. So yes, it did work at reversing fescue toxicosis. However, we're not going to use human supplements in animal production because it's way too expensive. So we had to think of other ways to be able to do that. So the first strategy we looked at, which is probably the most obvious, was interseeding red clover into toxic tall fescue pastures. And so we spent a couple of years and established a 30% red clover stand in our experimental pastures. And the animals were either grazed on just the plain toxic tall fescue or pastures that were interseeded with the red clover. And what we found was at the termination of grazing, the animals that were consuming red clover had greater vasodilation and also had improved average daily gains over the course of the grazing experiment. The controls gained about 1 pound per day, while the red clover treatment gained about 2 pounds per day. The next thing we were interested in was looking at other supplementation strategies for those that can't establish red clover. So red clover hay was what we used, and also 2 different levels to see if we could go a little bit lower and get the same effects. So again, animals either consumed a control pasture or they were given 15 or 30% red clover that was normalized for protein using dried distillers grains, which don't have isoflavones. And what we found is that both treatments led again to that vasodilation, and we saw that the average daily gain was effective at increasing at both levels. So through these experiments, we were spending a lot of time with producer groups discussing these results, and probably the most common question was, what about isoflavone source? I have white clover in my pasture. Will that work like red clover? And so what we did is we started a little study where we again went back to the pens with the goats, and we gave them 3 different potential legume isoflavone sources. We used red clover, white clover, and soybean meal. What we found is that with a fescue seed challenge, all 3 of these sources were able to cause vasodilation, so very effective. Probably the most important finding from this study was that despite the differences between these legume sources, they all were able to work. So for instance, white clover is very low in isoflavones in comparison to the red clover. However, it didn't take very much to get the same effect, and that's led us to believe that there might be other options for supplementation. So for instance, conventional loose minerals are used a lot in grazing systems, so we decided to try and see if we could add red clover to conventional loose mineral as an additive to be able to combat some fescue toxicosis symptoms. So we worked with Berkman Nutrition on this project to design it, and what we did is we had a 20% weight-to-weight red clover mineral treatment. So essentially it was clover, red clover ground into conventional loose mineral, and this was made to represent the white clover treatment in the previous experiment. We had a 15% and 10% treatment to see if we could go a little bit lower, and then we had a 0% control. What we found was that with fescue seed challenge, these steers were constricted as we would have expected, with no red clover. However, all 3 of the other treatments effectively were able to dilate the blood vessels and reverse that vasoconstriction. However, 20% was most effective. So it's important to note that in this study, it was just a pen study, so we were only able to look at vasoconstriction and not gain or performance. So it's important that we then are going to take this to the field, and we have plans with KBN at Eden Shale Farm to do another study with steers supplementing these mineral treatments to them in the field and seeing how that affects performance. So stay tuned for those results. So switching gears a little bit, in addition to fescue toxicosis, we as a unit work a lot on the development of antimicrobial growth promoters. When you think about antimicrobial growth promoters, you're probably most familiar with rumensin and Bovatec that might be included in feed or as a mineral supplement or a supplemental block in your pastures. So what is an antimicrobial growth promoter? It's an antibiotic or an antibacterial agent that selectively kills certain bacteria in the rumen. This alters how the nutrients are utilized and allows the ruminant to gain more weight or improve feed efficiency. So when we talk about antimicrobial growth promoters, it's first important to discuss the importance of the rumen. So cattle are unique in that their first site of digestion is this rumen organ, which is a huge fermentation vat full of many different metabolically diverse bacteria. They also have another site of fermentation in their hindgut. So when an animal consumes pasture, pasture material is full of many different nutrients. The microbial community in the rumen is able to break down many of these nutrients, as we have a lot of bacteria that can ferment protein or fiber or sugars into different fermentation acids. And these acids are then absorbed by the animal to meet their energy requirements. So thinking about it in a different way, we can compare the rumen to another biological system that we all know well, the pasture. So forage species are beneficial parts of our pasture, or something we want to preserve and enhance. However, we also have weed species that we use things like herbicides to remove to improve pasture productivity. Similarly, in the rumen, we have beneficial species like these fiber-utilizing bacteria that would be equivalent to forage species in the pasture. Then we also have wasteful species that are equivalent to weeds, like these methanogens here, who are producing methane, that we want to get rid of with antimicrobial growth promoters. So another group of wasteful species are bacteria that utilize protein, peptides, and amino acids. And those are called the hyperammonia producing bacteria in the rumen. When an animal consumes protein, the protein makes it to the rumen, and these HABs are able to break it down and produce ammonia. This ammonia is often wasted out in the urine because it exceeds the ability of the animal to utilize it. For this reason, we use antimicrobial growth promoters like monensin that kill off these HABs and are able to divert protein to the animal, where it can be utilized via enzymatic digestion in the stomach and in the small intestine. In contrast, we have beneficial species that we need to preserve in the rumen. So for instance, we have these fiber-utilizing bacteria. Enzymes that break down fiber are strictly microbial, so the animal cannot degrade fiber on its own, which makes arguably these bacteria the most important bacteria in the rumen to maintain. However, some antimicrobial growth promoters actually inhibit fiber fermenting rumen bacteria like monensin can. So that can be pretty detrimental in their application in grazing systems. For this reason, when we look at research evaluating things like Bovatec and rumensin, we see kind of a variety of results. Sometimes it works, sometimes it doesn't. And it's probably related to this inhibition of fiber fermentation. So another thing that we think about when we think about the use of antimicrobial growth promoters is the veterinary feed directive, which reduces the use of antibiotics in cattle production systems. Monensin and lacsalicid are still allowed to be utilized, but there's a lot more regulation on that. And for that reason, we think that antibiotic alternatives are of interest. So back to red clover. So as I had mentioned, red clover is rich in isoflavones, most notably biocannon A, and we found in our laboratory studies that biocannon A is a selective inhibitor to rumen bacteria, just like other antimicrobial growth promoters. So when biocannon A is added to a fermentation, it reduces HABs, or those protein amino acid fermenting bacteria that waste protein, and it promotes fiber utilization, which makes it a little bit different than other antimicrobial growth promoters like monensin. In consequence, we have increased fermentation acid production and likely increased energy. However, we needed to take it to the field to look at the effects on performance. So the first question, again, does it work? In order to look at that, we had 3 treatments. We had steers grazing a pasture-only control, steers grazing a protein supplemented pasture treatment, and then the same protein supplemented pasture treatment with the addition of biocannon A. And what we saw is that the addition of biocannon A increased average daily gain about 1/2 pound per day, which is very significant in grazing systems. So yes, it does work, but unfortunately, purified biocannon A is very expensive, so we cannot use it in our systems. So we moved on to look at other isoflavone supplementation strategies. So the first thing we did is we looked at 2 levels of red clover hay at 15% and 30% of the diet. One of the reasons why we did this was so we could monitor intake and make sure that they were getting the isoflavones that they required, and also because it would be extremely difficult to establish pasture treatments with 15 and 30% stance at red clover and consistently. So from this study, we were able to see that those red clover treatments were able to reduce those HABs, or protein wasting bacteria, and also improve fiber digestibility like we saw in the lab, which resulted in an increase in average daily gain. probably the most interesting result from this experiment was that the 15% treatment was more effective than the 30% treatment, and that the 15% treatment allowed for about an increase in 1.1 pounds per day, while the 30% treatment was only about 2/3 of a pound improvement per day. Another question that we wanted to look at was, what about different quality grazing systems? So sometimes we have high quality pastures, and in other cases our animals might be grazing more mature pastures or of lower quality. So what we did is we established 2 treatments. We had a high quality system that was with a moderate grazing intensity, and then we had a low quality system that had a very low stocking rate to simulate this, this very mature pasture grazing. And these were supplemented with red clover. What we saw is that the red clover was very effective at improving average daily gain to a protein matched control. In the high quality system, the DDG only supplemented steers gained about 2.1 pounds per day, while the red clover supplemented steers gained about 2.6 pounds per day, so about a half a pound improvement. In the low quality systems, we saw something pretty similar, from 1.7 pounds per day in the control to 2.1 pounds. So I spent a lot of time talking about research that we've done in backgrounding animals, but I wanted to switch gears a little bit and talk about a different aspect of our beef production industry. So Kentucky ranks 14th in total cattle with about 2.13 million head in the state. We are the #1 total cattle and beef cow-calf state and east of the Mississippi. When you break it all down, about 76% of Kentucky cattle inventory is beef cow-calf. And so we've done some research in this area. I'm going to share a few studies with you today, but also as a new scientist, this is an area that I'm planning to expand into. So the first study I want to share with you is a study that was recently completed at Eden Shale Farm with KBN and their program Beef Solutions, and it was to look at adding value to market cows. And so what they did is they purchased market cows from the stockyards and brought them onto the farm. These cows were then given or assigned to one of three treatments, either a endophyte infected tall fescue pasture control, a E+ tall fescue pasture that was treated with chaparral. Chaparral is a broadleaf herbicide distributed by Dow AgroSciences, which is now Corteva AgroSciences. This product contains 2 different chemicals. The first is aminopyrrolide, which acts as the herbicide. Then it also has metsulfuron, which is a plant growth regulator and actually inhibits the emergence of seed heads in tall fescue, which reduces their toxicity. We also had a 30% red clover treatment that was established using frost seeding methods. So the animals grazed these pastures for 63 days in 2018 and 2019. And at the end of that period, they were weighed and body condition scored and then processed through their Beef Solutions program. And what we found was that all the pasture systems, even for that short grazing window, were great for adding value to these market cows. Most notably, both the chaparral and the red clover were most effective. In 2018, we saw an added value of about $117 per head, and over that short grazing window, the cows gained 2.3 pounds per day and gained almost an entire body condition score over the grazing period. In 2019, we saw very similar results and an added value of $192 per head, average daily gain of 1.6 pounds per day, and then again about a 1 body condition score. Another area of interest to our research team is beef calf microbial development. So I've spent a lot of time in this talk talking about the rumen and how important it is in an adult animal for fermentation and for pathogen defense and other functions. But what's notable is that when calves are born, they're really more like your dog or a human, and that they don't really have a rumen and they have an overly developed abomasum or true stomach. So over the first 6 to 8 months of a calf's life, it will develop into this adult gastrointestinal tract. And it does that by changing from a milk diet over to a high fiber diet, which stimulates this rumen development. So we don't really know anything about beef calf microbial development. There's a lot of research that's been done in dairy calves, but we do know that the microbial community is essential for reducing susceptibility to pathogens. The microbial community works in immune development. And then, like we had discussed in adult animals, it's also extremely important for feed efficiency and growth performance. And it would be easy to suspect that this calf microbial development early in life probably can dictate performance later in life. Also, based on this susceptibility to pathogens role, calf diarrhea or scours is the leading cause of death and profit loss for the first 30 days of a calf's life. And this microbial community and its ability to ward off pathogens is probably integral in preventing scours. So since we didn't know anything about it, we decided to just start with a fact-finding mission. So we performed a study looking at beef calf hindgut microbial development. It's a 3-year experiment, but today I'll just be sharing some of the preliminary results from spring and fall of 2019. We were lucky to have 7 producer collaborators in neighboring counties of Anderson and Mercer County. We worked with the University of Kentucky Beef Unit and their cow herd, and also Kentucky Beef Network's Eden Shell Farm cow herd. Over the course of that time, we sampled 41 cows and 45 calves to get 355 fecal samples total, and we collected those from birth to weaning. So for the sake of time, I'm just going to share with you some of our preliminary observations. First of all, we saw that spring and fall are very different. That's not surprising, as forage availability when the calf is born and for the cow is very different between those 2 seasons. We found that every farm was different. None of them looked the same in their development. We suspect that that's due to management differences, but could also be attributed to forage availability, as we observed some pretty dramatic differences depending on forage availability on those farms. We had some calves that over the course of the study had to be treated with antibiotics for one reason or another, and we found that almost in all cases those antibiotics inhibited that early development of the microbial community by as long as 10 days or more. So pretty significant changes. We would suspect that other treatments like dewormers and vaccinations also might have impacts like that, and so that would be part of some of the future research we'll be pursuing. Also, through this study, we monitored the cows periodically over the course of the experiment. And we found that the cow's rumen is set back pretty significantly post-calving for at least 15 days. But due to our sampling constraints, we're not sure exactly how long. This isn't too surprising, as we've seen that in a lot of other species, but could be very significant when we think about cow productivity and milk production. So again, something that we plan to pursue down the line. One other observation I wanted to share the graphs for was one of our producers had many sets of twins in the fall of 2019, and his management system was to take one of the twins off of the mom and put it on a bottle and leave the other calf on the cow. And what we saw when you compared the calves that remained on the cow, or the green lines, and the black line representing the calf on the bottle, is that the diet dramatically impacted the development. So on the left here, we have our fiber fermenting bacteria, and on the right, we have lactobacilli. And lactobacilli, you might be familiar with, is like a probiotic bacteria. It's often supplemented to help with pathogen defense or in animals that are under stress. So you can see here that in both cases, the cow raised calf was very well ahead of the bottle raised calf for development. So this is impactful from multiple standpoints. First of all, we rely on a lot of dairy research to dictate our practices in beef calves. As you can see here, a lot of dairy calves are raised on the bottle, and so this would lead me to suspect that we would need to really learn more about beef calves to be able to really know what our beef calves need to be successful. And also that diet generally is very important for dictating development. And that's something we should pursue. So this last slide, I just wanted to share some of the things about what's next. So as I had mentioned, we're going to be doing a growth performance study with steers on Eden Shell Farm with the red clover mineral treatments. We are currently doing an experiment, or have just finished year 1 of an experiment evaluating the role of isoflavone source, so alfalfa, red clover, and soybean meal supplementation on growth performance in grazing steers. We'll be looking at the reproductive effects of red clover supplementation in beef heifers and cows, as I had mentioned. These isoflavones are phytoestrogens, and so there is some concern that they would impact fertility, but also maintenance of pregnancy. So we'll be looking into that. We have a study that will be starting hopefully next year, depending on COVID-19, looking at the effect of adaptation on fescue toxicosis susceptibility in first calf heifer production systems. Again, getting back into more reproductive impacts. Additionally, with this experiment, we're going to be looking at the impact of this tall fescue on early calf microbial development and their susceptibility to fescue toxicosis over time, as we've seen that there's a relationship between rumen fermentation and susceptibility to fescue toxicosis. So hopefully we'll learn more about that with that experiment. And then lastly, we're hoping to look at a study to look at the effect of forage quality on calf performance in gut microbial communities and beef cow-calf pairs and conventionally raised dairy calves to answer some of those questions we were talking about with those cow-calf results. So with that, I'll take any questions virtually, or feel free to contact me at my email address there below. We're always, like I mentioned, relying on our collaborators and producers to share insights and experiences that they have and concerns and questions they have. So please reach out if you feel the need. Also, we have this ongoing cow-calf research that's happening, and we are still looking for more producer farms that might want to participate in that project. So if you are interested in having your cows and calves traced by our research team, feel free to contact me at my email address. Thank you for listening, and have a good night.